Hello, welcome to today's meeting. Today's meeting will be a recap of some things that we've been talking about in past weeks, and I'm also going to review two different topics today. I'm going to talk about embryo energetics, and I'm going to talk about experimental evolution. This will be a lot of fun, a lot of papers to go through, but we'll get through it. So let's take a look at some of our things that first thing we're going to talk about is our special issue of cells. And let me go to that. So this is a special issue. The title is Approaches to Developmental Network Structures. And if you've been watching us recently, we've talked about this, that we've been trying to find a list of topics, a list of potential papers, and then we've been trying to solicit authors. And this is, a lot of hard work because there are going to be a lot of rejections, but there will also be some acceptances. And one of the things we need to do early on is to define the table of contents. And so this is something that is, I've just given the outline here, sort of the headings, the topics that we're looking for, and then a number of slots. And these slots can be moved around, so we don't know how many papers we're going to get in each topic. But I'm going to go through this so that people understand what the structure of this is going to be. So we start with the introduction, and it's usually some introductory article that kind of goes through the articles that are in the special issue, but also, in this case, it would be a primer on developmental networks. So it would be basically the first part of that article would be a recap of all the things in the issue, and then we'd have this primer in the second part of developmental networks to kind of go through what we mean to prime the reader for some of these different types of network structures. Then we have another section, types of developmental networks. So in this part, we would have a number of papers devoted to different types of developmental network. How have people instantiated networks in development? And these could range from, molecular networks to cellular networks to even social networks or physical networks. And so there are a lot of different potential topics to explore here. But, we'd like to have like one paper from each area that we can identify. We've done this. I'm not putting in like potential titles because I'm still working on that. And I didn't want to bias people if they have ideas, bias them in any one way. The next section would be on like a special topic. Actually, the next two sections would be on a special topic. And so the first special topic would be networks and tissue formation. So this would have a lot to do with networks in the formation of tissues. So it would be like different types of networks in muscle or in some of the other tissues that are formed in development. And, they could be physical networks. They could be other types of networks. Then there's networks undergoing change. For this section, we envision, something about like how networks interface with things like phase transitions or symmetry breaking. And, they kind of describe the dynamics of a network. So it could just as easily be a dynamical network. But we don't know exactly what papers we're going to get for this. So this, again, is going to be a shorter section, these two sections. This is going to have a lot of papers because we want to give a variety of different approaches in this section, in the second section here. And then at the bottom, we have the final section, using networks to resolve dilemmas. So we want to leave space for people who may want to apply a network approach to their problem, but they don't necessarily have that network approach worked out. So this could be like, some question in genetics having to do with phylogenies or gene networks, gene regulatory networks. It could be with like lineage trees and the network aspects of those. And by network, we're being very generous. We're saying it could be like a complex network. It could be a directed graph. But we want to give people a little bit of latitude so that they can come up with some pretty creative topics. So that's what we're envisioning for the table of contents. Again, I'm not showing the whole thing. I'm showing kind of the structure, overall structure. I have a copy that I'm not showing you that has proposed papers, but I don't want to, I don't want to bias people, so I'm just showing this version. But if you want to contribute to this special issue, please contact us, and contact me specifically, and let me know what you'd like to see or what you could contribute. I think this will be a lot of fun, the special issue. The next thing I'd like to talk about is Susan Crawford Young has been working on her ball microscope, and she sent me some pictures of canola seed that she put inside the ball microscope. So if you'll recall, the ball microscope is a spherical microscope with cameras mounted at different angles along the edge. And so there's, I think, 10 cameras at this point, and they're at different angles. So you put a sample in the middle of the microscope, and you have a lighting source, and then the cameras will record whatever's in that mount from different angles. And so this is what I'm going to show you right now. I'm going to show you a canola seed. She did a paper actually with her daughter in 2021 that she published in Biosystems. And that paper had some samples in it. It had some plant pollen samples. I don't know if she used the canola seed in that paper, but she sent me some more results from canola seeds, and I'm going to show you that now. So this is with the latest version of the ball microscope, so it has the latest cameras on it and everything. This is a 1 millimeter diameter canola seed. So this is something that she lives in the Great Plains of North America. So they have canola seeds. They can just grab it from somewhere and use it to do this. So the canola seed is held in place with a drop of water. You suspend the canola seed in water, and you have the microscope bottom end stage is microscope 1, side bottom microscopes 2 to 5, side top microscope 6 to 9, and top microscope is 10. So what that means is that you have this stage, and it's in the middle of this ball. And the ball has 10 microscopes, and they're mounted in the following way. The microscope bottom, or the sort of the thing that's going to show the bottom and the stage of the microscope, is microscope 1. So it's showing the bottom of the canola seed. The side bottom microscopes are coming in from the bottom sort of at an angle. So you're seeing sort of the lower half of the canola seed at different orientations. That would be 2, 3, 4, and 5. So that's from 4 different angles, 4 different viewpoints. Side top microscope 6 to 9. So this is again 4 microscopes around the top at an angle. And then 10 is from the very top, and it's symmetrical to 1 from the bottom. So you're getting, points of view at, I don't know what the angle is, but you're getting evenly distributed points of view on that thing. Now there aren't things around the equator of the canola seeds, as it were, but that's okay because we're going to use these images later and we're going to stitch them together. So there's going to be enough resolution, at least for decent resolution all across the canola seed. So this is, again, she's put the pictures in this document. So this is number 1. This is where you see it from the bottom. So you can see the stage, and you can see the seed through the middle here. So you can see the bottom. Then you can see the sides here. You're seeing from these different angles. And then this is the angle here. And remember, this is suspended in water, so you get some, this is the water here around it. And this is the light source, as you can see. And then this is from the top angle, so an angular from the top. So you can see this is a different side of the canola seed. And you can see that the illumination is making it look different, but it's just another angle. And so these need to be aligned. She's just sending me the images, and this is something that you're going to find. We've been talking in the meetings about an axolotl embryo at the 1-cell stage. And this is very similar. A similar problem is that, we have this background problem where we have to take the background out, but also that you have, they have to be aligned and then they have to be, reconciled at each angle. So this is going to be an image processing task. And then finally 10 is from the very top. So you're seeing downward towards the stage and the very top of the seed. So you can see here at all these different angles. Now the question is, can we take these seeds, and this is a very rough, I think probably would need to play around with how these are segmented out of the images and maybe even the placement of the sample itself. But this is something that's all doable, I think. And then, stitch this together and you would have a flat image of a canola seed from the one end to the other and around. And so this will be an interesting, we have some people working on it, Hari Krishna and Karan, so hopefully they have something that they would like to, maybe they can work with these data as sort of a test run. Maybe we'll step right into the axolotl data. So that's the hot new data for this week. Okay. So now we're going to talk about two sets of papers, and we're going to try to get through this. I'm not going to go in very deeply into each paper, but I'm going to talk about them. I find these topics interesting because they do deal with some of the things we talk about a lot in the group. So the first one is free energy of cell cycles and chemical oscillators. And I've been able to catch a number of talks on this topic. This is a topic that I'm interested in. This is a topic we talked a little bit about in the origins of the embryo paper, the first paper that we did. We've also been talking about the origins of embryos in evolution. So I think these works may fill in some of the gaps here. They don't get directly at some of the questions we were asking in these, but this is something that is definitely, I think, an emerging area, and it's something that's very interesting in light of those two research aims. So there was a book 75 years ago, well, I don't know how many years ago. Yeah, I guess it was about 75 years ago now. It was called What is Life? And it was by Schrödinger, the physicist. And Rob Phillips, who's a biophysicist, has written a paper here thinking back about this book at 75 years. So how is this book aged? What is it teaching us? These sorts of things. So Erwin Schrödinger, there's a quote that says, "These facts are easily the most interesting that science has revealed in our day." So this is about the intersection of biology and physics. And remember, at the time, people were kind of exploring this interface, but there wasn't like the kind of biophysics community there is now. There was very little data in terms of the physical parameters of things like the embryo or other systems. And so this was something that, you can speculate on and write a book about, and then it leads to other things. And in this case, it led to an entirely new field. So in this book, What is Life? Erwin Schrödinger was discussing how the dislocation of just a few atoms in a gene can bring about a well-defined change in the large-scale hereditary characteristics of the organism. So, this is like a very biophysical point of view, correct? You have just a few atoms dislocation in some, the biochemistry of a gene. And this can bring about a well-defined change. And I think he's referring here to mutation. And so this is a point mutation he's referring to. And so these are things, this was actually kind of predating our understanding of DNA in a lot of ways. So, you have to take some of the things in here with, in What is Life with a grain of salt, because they didn't really have the kind of knowledge we do now. So 2019 marked the 75th anniversary of the publication of What is Life, a short book described by Roger Penrose in his preface to a reprint of this classic as among the most influential scientific writings of the 20th century. In this article, I review the long argument made by Schrödinger as he mused about how the laws of physics could help us understand the events in space and time, which take place within the spatial boundary of a living organism. So there's a lot of references to space here, and I wanted to make clear what those were. So there are events that occur in space and time. And so These events could be something like gene expression. They could be something like morphogenesis. They could be something like behavior. All these things take place in space and time. But then, they're interested also in the things that take place within the spatial boundary of a living organism. So, depending on how you define that, it could be that the spatial boundary is, the edges of the organism, the edges of the cells in the organism. It could be the edges of the behavior of the organism. It could be the ecological range of the organism. So, there are a lot of things to consider here, but this is basically the province of biophysics. Through Schrödinger's book is often hailed for its influence on some of the titans he found in molecular biology. This article takes a different tack. Instead of exploring the way the book has touched biologists, such as Watson and Crick, as well as its critical reception by others such as Pauling and Perutz, I argue that Schrödinger's classic is a timeless manifesto rather than a dated historical curiosity. What is life is full of timely outlooks and approaches to understanding the mysterious living world that includes and surrounds us, and can instead be viewed as a call to arms to tackle the great unanswered challenges in the study of living matter. So we have this need to understand sort of the biophysics, and going back into these old books, it's often something that you find a lot of things that sort of inspiration for new questions, I guess. And so, this was something that, I'm not going to get too much into this because it kind of detracts from our main point here. But just to get into sort of this idea of accounting for the living organism. So the meaning of accounting for the living. So what Schrödinger proposed really was this accounting mechanism. And accounting in terms of physics, so in terms of like energy balances and in terms of different types of forces and other things. And it's certainly something we talk about in this group. But this idea of accounting for is really kind of interesting because, really in the mid to late 19th century, there was an explosion in our factual knowledge about the light given off by and absorbed by different chemical elements. Just as with our current proliferation of gene and protein names and the burdensome nomenclature for the pathways that connect them, the area of factual discovery in atomic spectra saw proliferation of fascinating and complicating spectral nomenclature. So this kind of goes into some of these, aspects of modern science where you have all these genes and proteins and names, and we need to find a way to bring these together. We also need to have this accounting mechanism for things, usually a quantitative accounting mechanism. And then that can actually allow us to do some of these other theoretical pursuits. So one of the outcome of these struggles in the 19th century was the discovery of empirical formula that gave a phenomenological mathematical description to measured wavelengths of various spectral lines. So this is in the physics domain purely, and what they did was they had this accounting of what was there, and then they used equations to figure out the relationships of what was there. And to get the equations, of course, is one thing. You have to figure out the relationships, but you also need numbers. And you need numbers. You need to know what the ranges of values are. You need to know what the parameters look like. And we can do this as simulation now. Back then, they didn't have computers or computational power, but to do the kinds of computation you might need to, say, simulate this equation. But you did have to have numbers. And so that's kind of where we're going with some of this other stuff here. So the next article is the free energy cost of accurate biochemical oscillations. So this is now getting into this idea of free energy cost. And so the abstract of this paper reads, oscillation is an important cellular process that regulates timing of different vital life cycles. However, in the noisy cellular environment, oscillations can be highly inaccurate due to phase fluctuations. It remains poorly understood how biochemical circuits suppress phase fluctuations and what is the incurred thermodynamic cost. Here we study three different types of biochemical oscillations, representing basic oscillation motifs shared by all known systems. in all the systems studied, we find that the phase diffusion constant depends on the free energy dissipation per period following the same inverse relation parameterized by system-specific constants. That's a lot of words, but that's basically the idea that you have that there's this sort of set of relationships here with respect to phase diffusion and free energy. And there's this relationship that we can parameterize and put numbers on. This relationship and its range of validity are shown analytically in a model of noisy oscillation. Microscopically, we find that oscillation is driven by multiple irreversible cycles that hydrolyze the fuel molecule, such as ATP. A number of phase coherent periods is proportional to the free energy consumed per period. Experimental evidence in support of this general relationship and testable predictions are also presented. So what they're trying to do is they're trying to approximate the free energy cost of different biochemical oscillations. It could be in the production of ATP, it could be in other domains, but we can actually look at a lot of these reactions and find that they're oscillations. And those oscillations have a couple of different sort of ways that they work. And so that's what they mean by motifs. And so you can use a network motif approach, which is to find like these small components of networks that define some of these relationships. So you're taking these relationships and you're using a network model and you're finding different motifs. So some motifs might be that they all come to a common point. Some might be that they are highly interconnected. There are different ways that you can represent the chemical reactions and things that matter in the system. So I don't know if they show any pictures of these motifs. But this is, so they go through a lot of different biochemical oscillations here. And they talk about how this is, they don't really have any, they do at the end here. So here's some of the motifs. This one here I think is A. Let's see if they have it down here. Okay. So A is illustrations of 3 network motifs for oscillation. So this first one is activator inhibitor. So you can see that R is influencing X, X is inhibiting R, and R is influencing itself. So this is what they mean by activator inhibitor. It's activating one but inhibiting the other. Then there's this one here called the repressilator. And that's where you have 3 things going on. They're all repressing each other. So it's a circular system of repression, and it's shutting everything down. We don't know what's coming into A, B, and C. It could be that there's free energy coming into each of them, but the interaction of B and C and A and B are all repressive in nature. So it shuts down, it inhibits B, no matter what's coming into it from somewhere else. And then C is inhibited by B in interactions with B, and then A is inhibiting B. So this is what I guess it's like a festival of repression or something, repressilator. And then third is the substrate depletion motif. So this is where you have S, some things coming into S, and some things coming into P, and they're both excitatory or they're both activatory, I guess. And then P goes back. I don't think it goes to S. It's not a direct feedback, but it's feeding back to this connection between S and P. So this activator between S and P is being enhanced by the activity of P. So it's a positive feedback, but not directly on S. And then there's this output of P. So this is the substrate depletion. So this is where substrate is being depleted here as it's going forward, and the feedback is here. So these are the 3 motifs, and this is what they've extracted from different natural systems that they've observed and modeled out like this. So we have these 3 different motifs. And so this is one example of how this can be done. If we go to this paper here, which is measuring now we shift to embryonic development. And we see that this is actually Rob Phillips and Peter Foster and Manuel Razo Mejia. And Rob Phillips is at Caltech, and I think they're also at MIT. Some of these people were at MIT. And so the abstract here This is one of these papers that previews another paper. So we're going to talk about the other paper, but this is the one that kind of previews it. What are the thermodynamic costs of development? In this issue of Developmental Cell, and this is a Developmental Cell paper that's in this issue, Rodenfels et al. Demonstrate that the high energetic cost of coordinated cell division that is regulated by phosphosignaling gives rise to a measurable periodicity in the heat dissipated during zebrafish embryogenesis. So they're looking at zebrafish embryogenesis, and they're saying that this there's periodicity in this heat dissipation due to coordinated cell division. So basically, there's energy being put into cell division. Cell division is occurring. It's regulated by phosphosignaling, which can regulate its rate and it coordinate when it happens. And this gives rise to when you measure the heat dissipation of the system, you can see a measurable periodicity, which makes sense because if cell division is coordinated, it's going to occur in waves. And so one of the reasons why we care about this is because it serves as a proxy for looking at how much work is done biochemically in cell division. So the paper we just saw was that there are these biochemical reactions going on, and there are these networks that are occurring, and these networks are driving cell division. So cell cycle, for example, is cyclical. It's kicked off by a series of molecular events. It's maintained by a series of molecular events, and the timing of that can be shifted in different ways by biochemical signaling. There's some free energy that's used for this, and so if you measure heat dissipation, especially in something like embryogenesis, where we know kind of how that cell division is going to be occurring a lot, and it's going to be occurring maybe in a coordinated fashion, this actually allows us to look at this periodicity. And so they talk about this in context in this article. So they kind of show this example here where you have signaling that involves sort of setting the cell cycle, regulating the cell cycle. You have protein synthesis and DNA replication, and these things are all occurring at certain time scales. And then this is their heat dissipation rate graph versus time. So this is time in the embryo in seconds. This is the heat dissipation. It's nanojoules per second, I believe, and it's going upward over developmental time. There's a little lag here, but then it goes upward. And so you can see that there's each of these things that are going on incurs a free energy cost. So, it's hard to measure this like a single thing happening, but when you can get this sort of coordinated activity, you begin to see this sort of how this works, and you can get a handle on the numbers. Okay, so then this next paper, I believe, is the paper. This is a different paper on this, so I'll go to this paper. So this is the paper in Developmental Cell. Heat oscillations driven by the embryonic cell cycle reveal the energetic costs of signaling. And this is by Jonathan Rodenfels, Carla Neugebauer, and Jonathan Howard. And so this is where they're measuring the energetic costs of early zebrafish development using isothermal calorimetry. So they're looking, it's an optical method where they're able to visualize the thermal values here. And calorimetry is basically how many calories or kilocalories are being burnt. Calories are actually a standard measure of energy consumption. So this is something that it's an older method, but this is something that you can see. So you have these different things, these molecular things that they're measuring, and then they have this heat flow graph where they're showing heat flow over time, and then they're looking at the micromolars of ATPs in the system, and they're able to look at this. They're able to generate this graph. So this is one of these graphs with two y-axes. The heat flow in nanojoules per second is equivalent to a similar scaling of micromolars of ATPs per second. So this is this just kind of shows you over the cell cycle what's going on here. There's mitotic entry, mitotic exit. So they're able to identify these different phases of the cell cycle. So they're able to do this real-time, non-invasive measurement of the energetics. The heat flow during the cleavage stage of development oscillates in phase with the cell cycle. So they're just looking at the cleavage stage of the zebrafish embryo. So they're able to isolate a certain period of embryonic development, and probably it's the one that's most likely to yield a clean signal. But we still, we can look at this with respect to the cell cycle. The CDK1 cyclin B1 phosphorylation dephosphorylation cycle, which is a major component of what regulates cell cycle, drives oscillatory heat flow. And then substantial energetic costs are incurred by cellular encoding of cell cycle timing. So these are all things they found. And so I don't know if they have any images here we can go to. This just shows sort of like the way that the experimental setup worked. And you see this graph again that we saw in the other paper. This is heat flow versus time, and then you have micromolars of ATPs per second. And that's on a different scale, but that's correlated with that. So you see the trend, and then you see the oscillation. And you also see this component of noise. So, there's this noisy aspect of it. And we talked about noise in the first paper as sort of a thing that's inherent or intrinsic to biochemistry. And it's something that maybe we don't think enough about in modeling developmental systems. But noise is ubiquitous in these type of systems. And, modeling noise, measuring noise is sort of, you could take a time series and you can take noise components out using something like a frequency domain analysis of some type. But then, you can also model noise using different methods. And so I think we've talked about like Brownian motion and what they call super diffusive methods, which are Brownian motion plus a little bit of non-normality, where you get like these spikes that, deviate from the from Brownian motion. So these are things that, you can model noise in different ways. And I'm not going to talk about that today, but just assume that like noise is a major component of this as well. And it's not a bad thing. It may be part of the measurement, but it's important to recognize that noise actually drives a lot of this. That noise and heat and noise and energy and noise in a lot of things drives the system. And sometimes it can lead to adaptation. Sometimes it has other functions. Sometimes it's there because you have all these components that are working together, and as a consequence of sort of their working together, sometimes they can't synchronize completely, and so you get this noise component. There's some people that argue that noise is a fundamental component of biology and that it drives a lot of biological function and innovation, but again, we're not going to get into that today. So they have some nice plots of heat flow oscillating with embryonic cell cycle here. They actually do a nice wavelet analysis with period versus time. So this is a nice way to plot out. You can take a time series and you can find wavelet components. So you look at the different periods and you can look at the power spectrum for these different wavelets. The idea of a wavelet is you take like a time series and you decompose it into different periods. You bring it down into, you recursively sample it into smaller and smaller windows. So you get like from a large number of periods down to like 64 periods. So you do this at like multiples of 2, I believe, and you get this sort of power at different scales of the time series. So you average it at different scales, and or you transform it at different scales, and you end up with these different what they call wavelets over time. So for each of these, you have different periods. You have like 512 period of 512, period of 1024. They're basically transformed, or I wouldn't say that they're averaged, but they're smoothed to a certain degree. So to a certain magnitude, and then each of those has a certain power. And so as you can see, the 1024 period band actually has a lot of variation in its wavelet power, which means that there's a lot of power in some of these bands here at 2000 seconds. And so, there are ways to like analyze these oscillation cycles using wavelets. And so this is a nice study. They can really get down and dirty and measure a lot of things here. Get some good numbers on them. So this is all great work. And there's a final paper in this group that I wanted to talk about. And this is also from Jonathan Rodenfels and some other colleagues. Jonathan Howard's on this. Carly Neugebauer is also on this again. This is contribution of increasing plasma membrane to the energetic cost of early zebrafish embryogenesis. So in this paper, they actually talk about the increasing plasma membrane. And so this is something in embryos where you have the size of the membrane as it's increasing, and it contributes to the energetic cost of embryogenesis. So the abstract reads, how do early embryos allocate the resources stored in the sperm and egg? Recently, we established isothermal colorimetry to measure heat dissipation by living zebrafish embryos and estimate the energetics of specific developmental events, and this was largely in cell division. During the reductive cleavage divisions, the rate of heat dissipation increases from around 60 nanojoules per second at the 2-cell stage to around 90 nanojoules per second. And so this is an increase at the 1024 cell stage. So this is like, early in development from 2-cell to 1024, and there's this rate of heat dissipation that increases. And so why is that? It's not necessarily due to cell division, because we know that cell division is probably, in this stage, we know it's somewhat coordinated. So we don't know exactly why it's being driven. It's not irregular. It just increases. Here we ask which cellular processes drive this increasing energetic cost. We present evidence that cost is due to the increase in the total surface area of all the cells in the embryo. So as the embryo expands in terms of the number of cells, it can expand in size as well. A single egg is going to be much smaller than the embryo ends up being. So, this is different in different organisms, by the way. But the embryo volume is going to change size, and it depends on the phase of the embryo. So first, embryo volume stays constant during the cleavage stage, indicating that the increase is not due to growth. So they actually rule out growth as a factor here. Second, the heat increase is blocked by this drug here, nocosidole, which inhibits DNA replication, mitosis, and cell division. This suggests some aspect of cell proliferation contributes to these costs. So this is actually cell proliferation, and it's, related to cell division. Third, the heat increase is in proportion to the total cell surface area rather than the total cell number. So this is actually not due to actual cell number, it's that there's proliferation and there's surface area that expands. And then this is, so in other words, you have this, the embryo is a certain size, and in this stage it doesn't increase in size. But what happens is that each cell, divides and divides and divides. And so there's a lot more cell membrane because you have more cells than you had before. So that cell membrane is the thing. It's not the size per se. So this is what they've, so that was third. And then fourth, the heat increase falls within the range of the estimated costs of maintaining and assembling plasma membranes and associated proteins. Thus, the increase in total plasma membrane associated with cell proliferation is likely to contribute appreciably to total energy budget of the embryo. So that's all I'm going to talk about for this section. I hope you found that useful. I'm going to go through some papers now on experimental evolution. And this is kind of a shift from what I was talking about before. But several years ago, the lab actually did a paper on experimental evolution. It was actually myself. But this is something that was based on the interests of the group here. And so this is called Evolution in Eggs and Phases, Experimental Evolution of Fecundity and Reproductive Timing in C. elegans. So this was published in Royal Society Open Science. And it kind of goes through, it has a bunch of different, it has a wild type and a bunch of different defined mutants that were used here. and a number of genealogies were created. So what happened was you had like a population of these different genetic strains, and then there was some selection process where certain worms were selected out and passed to the next generation. A number of replicates were made, and then those were selected, and then those would be passed on to the next generation. And then there was an assay of how many offspring were produced from each individual that was passaged. So it was passaged from generation 1 to generation, I don't know, I think it was like 25 for most of the strains. Some of the strains didn't make it out to 25 generations, but which was notable actually. But basically, this is the idea. You have a population of worms that reproduce through selfing. Then you passage a small number of that population into a new generation, and then they have offspring, and those are measured, and then those are selected and passed on, and they're selected at random, so they're not selected by any particular feature. And then the idea is to go through and look at the different strains to see how they performed. And so you can see that the wild type is probably the best marker here, where you can look at the performance. And this was more or less a linear increase over 20 generations. I think this plot only includes 20. I think there were actually 25 total. But and then you can compare the rest of the strains to that wild type. So these AAK strains there was some energetic aspect to them that was, an energetic mutation. So the mutation impacted their ability to reproduce, and so this had some effect on, their ability to go through this experiment. There was a little bit less than the wild type, but the double mutant was actually better than the single mutant AAK1, and I think AAK2 is much less robust in its fecundity, its average population size over 20 generations. And then you had other mutants like DAF16, DAF7. These were alternate mutants that they had developmental mutations that can affect developmental aspects, and they didn't perform as well as N2. There were some that kind of died off here. These were double mutants, AAK and DAF, which didn't make it past 15 generations. This is notable because they just couldn't be, they weren't able to increase their fecundity. In fact, their fecundity fell to 0 as the experiment went on. So this is really notable. And these double mutants actually had both this metabolic defect and this developmental defect, or this developmental mutation. So this is something that was interesting in terms of the biology of these mutants. The idea, though, is that you have this, I don't know if I have an image of how the experimental setup here, but which I don't. But the idea is it's a very simple type of experiment where you get, you have this population at generation 1, you select from that population, you put them into a new setting, they generate their offspring. You select from that, you generate an offspring. And the idea is that you're just selecting at random, so you're not really imposing any sort of artificial selection on it necessarily, but you're looking at what happens when you keep passing on these individuals and restricting the gene flow of the population. So usually it's like one worm will have multiple offspring since they self. By passing on individuals, if there are genes or there are different mutations that might be deleterious, it might crash the population, it might increase the population. And so for these different mutants, there's differences there. And so you basically get a lot of genetic drift, but you also get this ability to evolve because you're not picking from the same, you're not necessarily picking from the same lineage. There are mutations that are generated, so it's not just like picking from the same well every time. So this is just kind of showing some more of the data here. So there's some caveats to this approach, but this is basically how this works. And C. elegans is a nice model for evolution, experimental evolution, because you can do a lot of things with it. Now, there's some papers on, not just on experimental evolution the way I showed you, but there are other ways you can do this. So there's a paper called Natural Genetic Variation in Multi-Generational Phenotypes of C. elegans. This is Marianne Felix is a person from France who does a lot of stuff with natural variation in C. elegans and some experimental evolution. In brief, this paper discovers that some C. elegans wild isolates become sterile after several generations at high temperature. So you can go through several generations at high temperature. And this is a trait that can be, not only can sterilize them, but it's reversible. So it corresponds to alterations in non-genetic inheritance phenomena. So you can actually look not just at the genetics, but the epigenetics. And in this type of model, it's very simple to set up. You just have to do worm culture, which is, it's not easy, but it's not the most difficult type of animal or cell or animal culture that exists, or bacterial culture, or whatever. But you can set up these conditions, and you can go out quite a while, because the C. elegans generation is something like 3 to 4 days. So you can spend a lot, you can get a lot data on a lot of generations. So this is a nice paper where they identify in the wild isolate MY10. So I showed you the in the last paper. I showed you an example of the N2 mutant. This is a variant of a wild type mutant. It's not N2, it's MY10. So there are different mutations in wild type isolates. They're not what they call defined mutations, where you backcross it and you make sure that it's always inheriting that genetic background. But these isolates can actually be quite useful as well. And this is something when they, analyzed the results, they were able to isolate this multi-generational inheritance phenotype. Now, this can be actually quite powerful, because you can actually look up to 14 generations ahead and ascribe it to a transgenerational transmission. So this is a paper, Transgenerational Transmission of Environmental Information in C. elegans. And this is using, this is where you have, so in this case, the environment experienced by an animal can sometimes influence gene expression for one or a few subsequent generations. Here we report the observation that a temperature-induced change in expression from a C. elegans heterochromatic gene array can endure for at least 14 generations. Inheritance is primarily in what they call cis with the locus, occurs through both oocytes and sperm, and is associated with altered trimethylation of histone H3 lysine 9, which is this marker here. And so, before the onset of zygotic transcription, expression profiling reveals that the temperature-induced expression from endogenous repressed repeats can also be inherited for multiple generations. So they have these candidate markers that they use, and they're looking at transgenerational transmission. And in this set of experiments, they found that they can look ahead up to 14 generations. So something can be inherited for up to 14 generations for high temperature memory. So in this case, you have high temperature memory. This is something where they're able to, this is kind of the technical aspects of the markers. And then this is where they raise the worms at 20C, and they're able to look at this over a number of generations. So there are also other opportunities in C. elegans experimental evolution. So this paper is sort of advocacy for mainstreaming C. elegans in experimental evolution. And as I mentioned before, it's a very powerful tool, potentially. It has a very short generation time. It's fairly easy to raise a large number of individuals. The genetic basis of things is pretty well worked out. So we know the genome, for example, and we know what the defined mutations look like. And it's very easy to do genetic screens. I mean, relatively easy. And so you have all these things going for it. So it's a very nice system to look at and probe. And, aside from that, there's a lot you can learn about experimental evolution. It's really relevant to a lot of animal species. So you know that, for example, we can do this in bacteria for many generations, but bacteria aren't necessarily analogous to, say, humans or other animals. C. elegans, on the other hand, has a lot of homologous genes. It has a lot of, has organs, it has a brain. So there are things you can look at in this process, reproduction. So you have all these things you can look at, and they have some, analogy to some of the mammalian systems that people work on. So this is something that, we're really interested in kind of using. Maybe it's a very easy way to look at some of these issues. There are also things like heat stress and other adaptive processes that are very easy to kind of probe and manipulate. So we can actually get some pretty good information about biological plasticity. C. elegans isn't really known for its biological plasticity, but there are ways to probe this in development. So this is something that, actually allows you to do a lot of really interesting things. So the life cycle is short and it's well characterized, and you have mutant alleles that you can look at, define mutations. You can actually create custom alleles using genome editing. So you can use something like CRISPR to create manipulations in the genome, and then you can actually understand not only that, but these extra chromosomal transgenes and other types of epigenetic inheritance using C. elegans. And it's been observed in C. elegans. It usually relates to temperature or food. So when there's a lack of food or the temperature is too high, there's a transgenerational response in C. elegans that's been characterized. So all these things can be done. And all these things are relatively easy to to see results with. So this is the power of using C. elegans. This paper, C. elegans evolution, or Canarhabditis evolution in the wild, I should say. This Canarhabditis being the genus, kind of talks about some of the natural history of Canarhabditis. So we not only have Canarhabditis elegans, but we have other species in that genus, and they're somewhat different than C. elegans. They can be different, and understanding that natural diversity is actually pretty important for getting a handle on what we're seeing with experimental evolution. So even just some of the things that we talk about in the group, like lineage trees and things like that, those are not necessarily typical of even members of its own genus. So they're worth kind of understanding the natural variation. And of course, the natural variation is very important to interpreting what you see in your experimental evolution experiments. So this paper is by Ash Cutter, and this talks about, some of the aspects of natural diversity you see in that genus. So there's a lot of stuff going on, even within C. elegans in the wild, even with some of these wild type strains that we talked about before. There's a lot of genetic diversity, and we don't really know. I mean, we use a number of different strains for the laboratory. We use the N2 strain, for example, as a wild type. That's a strain from nature, but it's also been backcrossed quite a bit, so it's not like at one point I actually was able to harvest C. elegans out of a backyard mulch pit or a mulch pile, and so I was able to take samples out and get C. elegans out of there. Those probably have a lot different genetic makeup than something in the lab. I didn't genotype it, but one of those N2 strains is going to look very different genetically. And then the defined mutants, the ones with specific mutations that have specific functions, those are usually derived from the N2 strain. So understanding a lot of this natural diversity is very important. And especially in the openworm effort, I think there's a lot of opportunity to think more deeply about that natural variation and how it might play a role in simulating the worm and simulating some of the things that we kind of take for granted in terms of this, knowing every cell and knowing every connection in the connectome. So this is something that's quite useful as well. You can also study the evolution of development, and so this is a paper by Rolf Sommer summarizing the evolution of development in nematodes related to C. elegans. So talking about changes that occur in development that come from sort of evolutionary change. So you get changes in development or changes in evolution that translate into changes in development. And there's an interplay there. If you were to do these experimental evolution studies, you could actually manipulate, perhaps, developmental mechanisms. There's a lot of interplay between the production of offspring and that larval period, that larval stage, and there's a lot of biological plasticity in that larval stage that can play a role in, how you select adults or how you select individuals to produce offspring. So there are different ways that can be set up. And of course, needless to say, there are a lot of developmental changes that might play a role in some of these epigenetic changes as well. And then finally, one of the things that we like to think about with C. elegans in terms of medical research is the lifespan or life expectancy. So a lot of people have studied C. elegans to understand aging in humans, but in general, some of the genes involved in aging are homologous to some of the genes that we find in humans. And of course, you can see, with a lifespan of several days, it's very easy to look at the lifespan over and over and over again, and even look at how it evolves. And so this paper is about the evolution of lifespan in C. elegans. And so it was proposed almost 50 years ago that aging is non-adaptive, and is the consequence of a decline in the force of natural selection with age. So this led to a theory that aging results from detrimental effects late in life of genes that act beneficially in early life. So there's a theory that people had kind of come up with in C. elegans. Using C. elegans, this group showed that a mutation that greatly increases the lifespan of the nematode C. elegans does indeed exhibit a fitness cost, as demonstrated during starvation cycles that may mimic field conditions, thereby validating the pleiotropy theory of aging. So this is something that is sort of this thing that they proposed 50 years ago. They're actually using C. elegans to sort of find evidence for it. And so in C. elegans, you can actually starve them pretty easily in development. You can do L1 starvation, which is where you take the eggs and you harvest them and you place them on a plate so you can synchronize the population. And you actually don't place them on a plate right away. You put them in a tube in buffer for about a day or two. You can keep them, I think, for up to 2 weeks. And this starves the eggs. It kind of gives, sort of sends a starvation signal to the egg, to the cells. And so when you plot them and you put them on a plate and you let them start to go into their larval stage and hatch, they'll rest in this L1 stage in the tube. And so you plot them out, and there are these L1 worms, these small worms, and they kind of stay in that stage for a while, and then they start to grow. But then they also exhibit changes due to that starvation experience, and it affects the way that they sort of mature. And so you can do these starvation cycles, over multiple generations, and you can actually force a certain adaptation. So you can observe, for example, these epigenetic mutations or these epigenetic changes. You can observe, maybe you can select for mutations that favor starvation environments. So there are different things you can do. These starvation experiments are very well characterized in the literature. But combining that with experimental evolution can give you really interesting results like this. It can actually address some of the issues in the aging literature, and maybe some of the phenotypic plasticity literature as well. So they were able to actually identify some alleles here, some genetic alleles that were involved in aging, and this is an interesting little communication here. The observed fitness costs associated with field-like conditions, which means that it would be analogous to something you'd find in nature, provides an example of a single gene that acts early in life. Aging and Darwinian fitness is predicted by the pleiotropy theory of aging. So that's pleiotropy is where you have a gene that has multiple effects. So, this is something that people have proposed, and they were able. This is actually goes back to 1957, an old paper by G.C. Williams in Evolution. So this is, again, something that was published in the year 2000. And it took them 50 years to find evidence for it. So this is a nice set of papers. I hope you enjoyed this session, and see you next week.